Have any of you heard me speak before, so I won't be redundant? Oh, good. Just a couple. The product is launching, as Alan said, launched a couple days ago, but formally next week in Utah is the ageLOC True-Face Essence Ultra. It's a Generation 3, iPhone 5 as you say, the TFEU that launched in 2003 when the company for which I served as Chief Operations Officer at UCS Pharma licensed the rights to marketing exclusively MLM to Houston. And these slides are going to be available on.
Yeah. Yeah, so we'll give it to one of the leaders and let's leak it out to you guys.
So no copious notes, you can sit back, relax, and know that. And I'm not going to read each of the slides because that's tedious as well. Basically, as a summary slide, it's slide number 2, and there are 69 slides, so relax. I have to catch a plane in, well, I have to leave for a plane in an hour, so it won't be longer than that. So ageLOC TFEU is a novel and proprietary formulation. New Skin with its ageLOC technology of upregulating and downregulating genes, and our company came to this new formulation such that we actually had the evidence in the gene technology using proprietary to New Skin to prove the upregulation of both elastin and collagen synthesis. Ethocyn, the ingredient to which I am the inventor, is a topical clinically proven bid regimen. It's twice-a-day application, morning and evening. It's very safe, although in all these studies that validate and substantiate these claims, target tissue, which means it only acts at the site of application, on the skin, non-steroidal cosmetic ingredient. Again, this is sort of a summary slide as to all that will be presented to you here this afternoon. And New Skin asks me to tell you a little bit about my background. My first love is biochemistry. My father invented the milling machine gun. For those of you who know, when you build a house, you don't only use hammers and nails. He has 2 daughters, my sister and I, no son, so I became the son. He was born, and he asked me to leave my biochemistry studies at UCLA and become an attorney so that I could one day take over his world steel companies. He vertically diversified into the steel industry after inventing the machine. The patents expired after 17 years, and then he bought steel mills and drew wire per rod to make mills. That's just why I became an attorney. And in fact, I did join him in return in his company until he sold into Westinghouse. But he asked me to leave biochemistry because he said the probability of my inventing what I had already started envisioning, which was a non-steroidal anti-androgen. He said, you know, Chantal, the pharmaceutical industry has wall-to-wall chemists in research, and maybe after 50 years they'll pat you on the head, give you a gold watch. And he said, I would really like you to come work into the family business. I will give you a laboratory. And it was in that lab that Ethocyn and its over 10,000 other analog structures in the patents came to existence. I now serve as Chief Operations Officer of a pharmaceutical company headquartered in Singapore. My family is in Los Angeles, where I reside with my 2 daughters. This presentation is these 4 bullets. I'm going to go briefly over the research and development of Ethocyn as a molecule, the $20 million plus of safety and efficacy studies just to the Ethocyn's mechanism of action in clinical trials. People ask me, what is Ethocyn? It's a powder. It's a clear, colorless oil that comes off this research bench, and now it's in synthesis in our laboratories. And its cost is over $100,000 a kilo. That's a very expensive cosmetic ingredient. But there's no allocation of G&A to that 100,000 kilo cost. That's just for the raw materials, knocking everything over, and the chemist at the bench. So New Skin, when they charge you this price, it's not because they're trying to make a lot of profit. It's because they paid a high cost of goods for this ingredient. The cellular physiology, for those physicians and chemists in the audience, and depending where Alan and other New Skin corporate people ask me to present, my presentations are either more or less technical. So I beg you to bear with me on those slides that are too technical, but at least you'll have the feeling that you know what Ethocyn's doing mechanistically. Ethocyn competitively blocks the intracellular DH2, which stands for dihydrotestosterone from binding to the dihydrotestosterone receptor. The testing has been worldwide in laboratories that are independent labs, and many times cosmetic companies will tell you that they have an ingredient that's proven to do X, Y, or Z, but what they've done is they've tested that ingredient in their own company labs. What you want to ask people when they're asking you to sell a product is the testing independent? And Alan has been talking about the plastic surgeons and dermatologists that he speaks to. That's one of their first questions. Is this testing in the company labs that nobody can go behind the closed doors and understand the protocols, what's happening? This ingredient, Ethocyn, was first presented to the FDA because being a non-steroidal anti-androgen, I wanted to make sure that it was safe and efficacious. And we filed the IND with the FDA and all the preclinical studies. The FDA recommended these labs around the world. We know now that Ethocyn is for all skin ethnicities. Males and females are born with DHT receptors, and they benefit, return to the elastin content of 20-year-old skin after the twice-a-day application for 45 to 60 days. And anyone over the age of 25 years needs to use this product because their fibroblast cells are making less elastin than that of people under 25. Patents have issued, as I mentioned. New patents are pending around the world. They are in 3 categories: composition of matter, process and use patents, trade secrets, exist for the isomeric compositions, and the trademark is for Ethocyn. Scientific publications. Second question asked by dermatologists and plastic surgeons is, are there publications, or is this a company just saying our product does this, increases elastin? And the publications are peer-reviewed. They're some of the most prestigious medical and scientific publications in the world. They're available to you. You can ask your lead blind person. There's also a clinical brochure with all the preclinical and clinical studies. It's listed as a cosmetic. Again, we can do a Q&A.
Yep.
Many times I'm asked, is it a drug or is it a cosmetic? Because of the safety studies and the proof of no untoward side effects, no penetration into systemic circulation, Ethocyn has been classified. It is a cosmetic. It's listed in the International Cosmetic Ingredient Handbook. This is the 2012 version, and also in Europe in the Cosme Dictionary. Structure, any chemist in the audience, it's a bicyclooctane ring, 5-carbon side chain. Lipophilic penetrates, molecular weight 266. So it's unique because it's the only ingredient that is known because of the comprehensive patents in all 3 categories to increase your elastin production. And I could go on about elastin and other products. Maybe we'll do that in the Q&A versus in this presentation on the front. So what does having more elastin fibers do in the skin compared to a moisturizer or an exfoliant? The elastin fibers are the rubber bands in your skin. Your skin is comprised of 2 forms of fibers, collagen fibers and elastin fibers. Your scar tissue is collagen. It's structural, it's dense. The elastin fibers, which intersect the collagen fibers in the matrix, are the rubber bands. So when you scrunch your face, or you frown, or you lift the back of the skin on the back of the hand, the bounce back, the snap back of the skin are the elastin fibers, the rubber bands that give you the resiliency. With age, and we know this from the pinch biopsy analysis, somewhere between 25 and 30 years of age, every individual, their fibroblast cells make less and less of the elastin protein. Therefore, you on pinch biopsy will have less and less elastin fibers, less rubber bands. Skin starts to sag, it starts to wrinkle soon thereafter. There are 3 forms of skin aging. I'm sure you're familiar with this. Mechanical aging, extrinsic and intrinsic. This product, in particular the Ethocyn in this product, there are other ingredients in the 21 ingredients contained in the TFEU ageLOC that do other things, but the Ethocyn itself, because of increasing the elastin production, targets intrinsic aging, the aging that occurs genetically because the DNA codes for less elastin production. Dr. Peter Puglay is an MD and eminent skin physiologist and a colleague of mine that we collaborate on studies. Has a medical textbook that he has written prior to his knowledge of Ethocyn called The Physiology of the Skin. It's available to you Chapter 11 is a wonderful chapter for those who want to read previous science. That elastin is the youth protein. It's the main reason, the loss of elastin, that your skin sags and wrinkles. This is an excerpt from Dr. Puglosi's book. It's also one of my slides for next week's presentation, the skin shows, which talks about how wrinkles form, why. If your mother wrinkled in the middle of her brow, you'll wrinkle there. And that's because of the position of the muscles under the skin. It's not, but all of you, when you start losing the elastin production, you will start to form wrinkles either symbolic or representative of how your mother or father wrinkled. So some cosmetics say they contain elastin. And if you go to someone and say, here at U-Skin we have this wonderful ageLOC product of TFEU, they'll go, oh, we already have a product. We know a product that has elastin in it. And what you need to say to them is, no, this doesn't contain elastin. This contains an ingredient called Ethocyn, and that's a trade name that I filed with the FDA as well as the IUPAC, the National Bureau of Nomenclature for Chemicals. Ethocyn means that the elastin is synthesized. So that was the derivative of that particular trade name. Elastin is a molecule, as I mentioned, in the skin of the fibroelastin. Its molecular weight is over 200,000. That's a huge molecule. So if you put elastin on your skin, as many cosmetic products contain elastin, it just sits on top of the skin. The reason that the FTC, the Federal Trade Commission, can't take action against companies that sell you elastin is because if the claim reads, if the copy on the advertising reads, our product contains 92% bovine collagen, elastin, and your skin will look younger. True statements, all three of those. It contains the bovine elastin, I think collagen because it's our product, it contains collagen. It's 92% pure, it contains the elastin molecule, and the skin looks younger because elastin has the ability to hold water to it. So it acts as a moisturizer. But it doesn't go in the skin because of its molecular weight. And if it did go in the skin, it would just sit in there as an elastin molecule. It's like a bald person that would take a product with hair in it and put it on top of their head. They're not going to have better hair because it doesn't integrate into a hair fiber. So you want to have Ethocyn applied topically so that your own fibroblast cells make your elastin protein and fibers. So Peter Puglose has made this quote at medical conferences, and it is, the first statement is from his prior work with elastin, that the single most important cause of age-related wrinkles is the loss of elastin fibers. You can move forward. And then the last line, only one product in the world can restore your elastin levels to that 20-year-old skin, and that is Ethocyn. This is a mechanism of action I'll go into in a few moments. So both men and women are born with DHT receptors in the cytoplasms of your fibroblast cells, other cells in the body as well. You contain DHT receptors from birth. We are genetically predisposed to inheriting the amount or quantity of DHT receptors that we possess in the different DHT receptor-containing cells. So if you take a child, scientists, if we take a child and do a biopsy, we can measure by retrieved hydrogen assimilation counters how many of these protein structures called DHT receptors they inherit in their fibroblast cells, which are responsible for making elastin, in their hair follicle cells that cause the hair to grow or stop growing, in their sebaceous gland cells, which produce sebum oil for acne. So an anti-androgen, and those of you who are in the science world, has many therapeutic indications. Our company, a pharmaceutical company, is processing through the FDA indications for this class of compounds for androgenic alopecia, male pattern involvement, acne, keloid scar tissue, hirsutism, women that have facial hair. You may question, well, do some people have, age wrinkle younger or later than others? African Americans, for example, have fewer genetically. They have fewer DHT receptors in their fibroblast cells, so they tend not to have loss of elastin until much later than Caucasians. Asian population, I'm talking genetic populations, have in between the two, so they also age later than, in terms of skin laxity, than the Caucasians. Male pattern baldness, if you have too many DHT receptors in your scalp, in the frontal area or the vortex area, you will bald in those areas. Take biopsies of a child's scalp, you'll know if that child will bald once they pass puberty, when the hormones turn on and the binding starts occurring. Middle Eastern women, for example, very often have hirsutism, facial hair. In the clinical trials we do, we've seen them with facial hair on the chest, the stomach, the pubic area. Do a biopsy, they have many DHT receptors in those skin follicle cells. This is a graphic. So what we have here is the DHT receptor that we all contain cytoplasmically in the fibroblast cell or other cells. DHT loves to bind to its DHT receptor. Again, eunuchs and castrated men don't have these disorders because they don't have the circulating hormone, but they have the DHT receptors. Ethocyn I designed with a molecular affinity for that same DHT receptor at the 5-beta, at the 5-ketone position, the 17-beta hydroxyl position on the molecule. So Ethocyn doesn't bind, and all this DHT can't get into the receptor because, like on a football field, the Ethocyn's in there blocking it. And that's the very simplistic way to share with you how it works. It penetrates, as I mentioned earlier, its molecular weight is 266. It's organically synthesized, which is 11-step organic synthesis. Any chemist here, when I showed you that structure, the bicyclooctane ring, would just have put their hand to their head and say, difficult synthesis, that's the expense. Behind it, it's got Grignard reactions, diazomethane reactions. Penetrates C14 and H3. Doctors say to me, Chantal, would you please give us the clinical evidence, the studies that you have C14 penetration? Because many cosmetics, and say they do things, and in vitro studies, in laboratories, you can get fibroblast cells or skin cells to do things, but when you put something on topically, clinically, as soon as it's applied to the skin, your body's pH, the enzymes in your skin very often deactivate. So we needed to prove that we penetrate to the dermis and we stay active, and that's the beta oxidation slide that I'll show you a bit later. Here's a more macro view of what's happening intracellularly. So the nucleus codes for the elastin protein, the Ethocyn's blocking the receptor in this fibroblast cell. So I think more important than all that skin physiology and mechanism of action is the clinical proof that it works. And you want to ask, if you go to a cosmetic counter, as I sometimes do in department stores, and ask about new products that launch, have there been clinical trials? Are they quantitative endpoints in those protocols? And we have done all of those with Ethocyn. This, for people who ask me also, where is it made? It's made in our laboratories in Germany. This is the laboratory facility. I mentioned it's an organic synthesis. So the clinical efficacy, Ethocyn is a clinically proven topical. You don't eat those little pearls. Although someone once asked me what happens if my baby gets it, and we did oral studies as well as topical in all clinical work, and there is no side effect. It's just an oil that people have a little bit of diarrhea after. Don't go eat. My children always say, Mom, you're so clinical. You don't talk about those things. I'm a scientist. I embarrass them. And neither one of my children, they're now 22 and 24, went into science. They're both I think they grew up with too much of this. We're going to do other things. My youngest daughter is going into environmental science and going into law school. My oldest daughter is going to law school as well. So they went into law. I did that too, but they did no science. Here we have the clinical trials protocols. They were very objective clinical trials. The p-value. Does anyone here know what a p-value is? Okay, this gentleman.
Statistical reliability has to do with it. The smaller the p-value, the more reliable the results.
Thank you. Did everyone hear that? It's a probability statistic. Exactly. So the probability that the product will do in the mass population of people what it was shown to do in the clinical trials that are either processing through the FDA or just in a company's portfolio of clinical trials. So you want to know that what we saw in our clinical trials will happen to you and everyone else that uses the product. An NDA, a new drug application or a new drug approval, requires in the United States a p-value of 0.05 or better. This p-value is 0.001, which, as this gentleman said, is less and far better than 0.05. That means When a p-value is 0.05, it means there's a 5% probability that what you saw in the clinical trials for safety and efficacy won't happen to you. A probability of less than 0.001 means that there's less than 0.1% that what I'm showing you here today won't happen to you and everyone else who uses it. So rest assured that your skin will return to that of, rest assured that's 0.1% probability that your skin will be, you will have the skin. There's a 99.9% probability your skin will be that of 20-year-old skin in the lasting content. Clinical trials at UCLA were our first studies. Dr. Richard Strick, they're a very eminent academic dermatologist, is the person I went to. I did my studies at UCLA. Dr. Strick was really high medical acclaim and sterling credentials. He's not one of the doctors that you put on TV on an infomercial. He's somebody who said, I want to study pharmaceutical products. I will study this product, Chantal. He was very material in the design of the protocol, which is very important because you may hear in the science world you can design a study to say what you want it to say. And we didn't want to do that. We wanted to see what the product does and how safe it was from a purely objective point of view. Dr. Strick presented his studies at the prestigious American Academy of Dermatology that year when those studies were finished, and he compiled his data, the conclusion being that only Ethocyn can give your skin the lasting fiber content in 20-year-old skin. Dermatology Times. This, Alan, also is a publication available to people who would like to read it in detail. This is what a biopsy looks like, and Alan asked me this morning, before I presented in Seattle, if I could give you a bit of overview of what it means to do a biopsy of the skin and how to prove that there's more elastin. So a 2-millimeter punch biopsy, you may have heard about it like in cancer. People take a biopsy of a tumor. It's like a stick with a very sharp end that goes in, punched into you, and it comes out with a piece of skin in the hole of the stick. That piece of skin, the 2 millimeters of that skin, is then put in media in a Petri dish with an elastin preferential stain called Burkhardt stain, which means that that black stain is only sucked up by the elastin in the biopsy sample. And then that biopsy sample is put on a microscope slide, put under a microscope. The microscope is hooked up to a computer screen. The computer screen scans, and this is what you're seeing here, are the scans, the amount of black. So the first side is this is a 42-year-old woman, her baseline, which means when she came to the clinical center, in this case it was UCLA, and took a biopsy before ever using Ethocyn, she had 3.3% of her skin were black elastin fibers. After twice-a-day application topically, for 2 months, another biopsy was taken, and she had 16.7% elastin. Of course, what you care about after that is, there were over 250 patients, their age range. This original study, as Alan said, was 40 to 77-year-old patients. We then studied down to 25-year-olds. Before Ethocyn, there's never been an ingredient that measured elastin, so we were going down a lot of frontiers in this research. Physicians didn't know how long it would take to increase your elastin fiber content. Alan mentioned that in this new ageLOC product that there are ingredients that have, galvanic, but also now there are 2 or 5 new ingredients that will give you an immediate effect versus having to wait to see the increased elastin after 45, 60 days. Because people sometimes say, I want to see it working right away. It's not the Ethocyn working right away, it's the other ingredients that we put in there that increase collagen, as well as cause some withholding of the life of the lipids in the skin. So there's some puffing and the wrinkle puffs up. That's how moisturizers work. And I'm not saying moisturizers are wrong or bad. You put a film of oil on your skin, the water can't escape under it, because there's a film, and the wrinkle puffs up. That's what it does. Simple. The world of cosmetics has been the world of moisturizers for many years. And it's not wrong to do that. You look better because your wrinkles are not as apparent. It goes away after a few hours. Sometimes you can see in the claims that many companies are moisturizers last 8 hours, 10 hours. So in this new formulation, we have extraordinary moisturizers, antioxidants, anti-inflammatories, collagen boosters, and inhibitors, but I'll be back. Just again, to independent laboratories, important BioQuant is an eminent biopsy, staining, and analysis laboratory in Tennessee. This is some of the slides they provided me to present as far as the equipment they use. Here are samples. I didn't show you the best slide, which some companies do. As I said, 99.9% of the people have the elastin content of 20-year-old skin. If you are born with very few DHT receptors in your fibroblasts,
Ask you to do one thing. I'm just going to put this on there. No, you can just hold on to this. Roughly. Oh, no.
Except that it's ringing. It's buzzing.
No, it won't bother.
It won't bother me.
Say hello.
That's the next thing. We'll be having a conversation. Thank you. It's much more comfortable. I was having a hard time.
can you hear me okay?
Yeah.
It'll also be recorded for you guys.
Thank you.
The man is marketing. Al and I are good friends. Without marketing, that's body safe in the laboratory. So I came to use Skin. It's the premier marketing company in the world. I'll comment to that a bit later as well after I get through a little bit of this time. So these are the biopsies of many patients. If you were born, as I was saying, with very few DHT receptors in your fibroblasts, then you would be in that 0.1% population where you wouldn't need the product. But almost everybody in the world loses elastin fibers somewhere between loses the production of elastin fibers somewhere between 25 and 30. The control group was 18 to 25-year-olds because it wasn't known what is the percent of elastin in young skin, because there'd never been a product to test for elastin. So at UCLA, they took a group of 18 to 25-year-old students, males and females. There were 20 students in that population, 17 females and 3 males. And their elastin content was about 15.6% on mean, on average. So there were those who had 14%, there was a couple of patients that had 18%, but the average for young skin is 15.6% elastin. Everybody that uses this product gets at least 15.6 to 21%. The safety evaluations, again, this is not a technical audience, a physician audience, but we did full hematology, serum chemistry, hormone assays, showing that Ethocyn is very safe. No one has to worry about it. Physicians, of course, ask that question when you're dealing with an anti-androgen, and this is the reason why. We look at we did C14 studies in Burnison in Holland, which is the only place in the world where they allow carbon-14, the radioactive isotope, to be put topically on people versus animal studies. And then you take a serum sample under the skin right after application, then you do urine and fecal examinations. There's no Ethocyn that ever enters the body, which is important for a cosmetic ingredient, of course, in anything. And this is because, again, there's beta oxidation of the 5-carbon chain and the non-anti-androgen primary metabolite is what happens. The downside to this, positive for marketing people, is that you need to apply this somewhere between 8 and 10 hours after the initial application, because that Ethocyn molecule gets metabolized by beta oxidation to the non-metabolite, so it doesn't sit in front of the receptor longer than that time. So you have to apply it again. We do Q&A as far as people ask me, can I apply it once a day versus 3 times a day versus double the dose. So the clinical implications of decreasing elastin is a tissue elastin will decrease in quantity and quality with age. Skin becomes less elastic. Skin loses its firmness and starts to developing wrinkles and new tips. Not readable, clearly these charts. This was the clinical results of the first phase 2 clinical trial at UCLA. The 6-month study, 3-month study, 40 to 77 years of age, males and females. For me, the average on baseline, which I mentioned to you, is when you come into the study not having ever used Ethocyn before. In the first 6-month study was 9.2%. In the second 3-month study at UCLA, it was 8.1%. Statistically significant, it was just that the baseline populations of those 2 studies and people that came in were different. So there were more people with higher elastin in the first study. At the study conclusion, the mean of the first study, it had doubled to 18.2%, and in the second study, it actually went almost 3 times to 21.3%. What's important is that after the study, we're in the 15.6% or higher, higher range of young skin. Ergo, the substantiation, the plastic surgeons, the doctors, the scientists say, please show us your clinical data. How can you at New Skin, or I, when I present, say that you return the skin to 20-year-old skin? It's because we did studies in the older patients, and they all had higher than the elastin content. Control group which was 18 to 25%. There are people that come in at baseline, and they have 3% elastin. There was one person is in Shanghai, they had 0.1% elastin. And these people have very saggy skin, very lax skin. We did their DHT receptor profiles, and they had many, many, many more DHT receptors than someone that came in, same age, let's say 40 years old, that had maybe 8%. Those people that come in with 1, 3, 4, they all go up to the levels of new skin. So it could be a 600% fold increase. These are charts. Again, you can using the PowerPoints when you have access to this PowerPoint. And this is the actual verbiage that Dr. Strick presented at the American Academy of Dermatology, the conference, the publication conference that he originally agreed to do the study. Over 10,000 physicians around the world attended the AAD, and he had a standing ovation when he presented the results. The standing ovation was very much to his credit for designing the protocol such that there were no rocks he didn't look under. So, for example, in Retin A, you may be familiar with the Retin A compound with J&J. It's somewhat of a discriminating, it increases collagen, but Retin A's first trials, Retin A is like an active product that then received an NDA, FDA approval for skin wrinkling. And Retin A's mechanism of action is that it causes erythema, swelling. So if you go out into the sun, if any of you have taken Retin A or put it on topically, you put it on topically, you will have redness and swelling. Well, that will make wrinkles look better. And so when Dr. Boris out of Michigan presented his Retin A trials back at the SIRD conference in France, where I attended that, many of the physicians in the audience raised their hand and they said, well, Dr. Boris, in your protocol we see that there were steroids applied for the inflammation, for the reddening. And steroids are known to treat skin wrinkling. All to say, this protocol was so pristine that no physician in the audience at that AAD had any questions of Dr. Strick to doubt that what was happening was because of Ethocyn and that it increased the elastin. Since those early trials, we do house many physicians around the world. This is Dr. Jeffrey Hoagland. He is the academic plastic surgeon, has a private practice in Beverly Hills. And the MTD stands for maximum therapeutic dose, because we had dose-ranging studies. Because, again, we didn't know how much of the Ethocyn percent-wise would cause this result. We did the first one, which was a very high percent did. We wanted to lower the dose so we could make it commercially available to the public. And so now Dr. Hoagland and I, last year and this year, are presenting around the world. You may read about this. So the medical journals it's published as Ethocyn being used as an adjunct treatment for laser surgery. So I wanted to give you a few of the slides today. Elastin's role in wound healing. This is his slide. His focus is to the academics of how collagen and elastin interact with each other and benefit in older patients. So lasers, you may know them in photon lights, that particular plastic surgery dermatology procedure is to increase the collagen, which also decreases with age in your skin. Dr. Hoagland said, Chantal, what would be wonderful is if we could do a study where we show that Ethocyn increases the elastin, which is very important, most important, and we use laser surgery to increase the collagen. So we're very synergistic with the plastic surgeons. Originally, our marketing people were saying perhaps the plastic surgeons would not be a proponent because it's going to take business away from them, and that's not the case. They are happy to have, for example, in postmenopausal women where the estrogen's off and there's no estrogen's a natural age to blockade. Postmenopausally, once the estrogen is no longer manufactured, there is marked wrinkling and skin laxity. Women that undergo hysterectomy, their skin very quickly will start to sag. So he was saying that we get a lot, plastic surgeons have a lot of women postmenopausal that come in for facelifts. And he said it would be wonderful if we could use Ethocyn with them 60 days before they have the procedure, so they have many, many elastic fibers. and then the surgery is optimized because the plastic surgeon doing the facelift has to compensate for the swelling that's going to happen. But if there's very few rubber bands, elastin fibers, they're then maybe pulled too tight to compensate for the, with an eyeball, compensating what's swelling is going to occur. Some of them look like they're men, look like they've been through a wind tunnel. So there are many, many plastic surgeons around the world that will not do a procedure without first applying TFEU with Ethocyn 60 days before so that the surgery is optimal. This is a fractional laser research document. This is the protocol to Dr. Copeland's adjunct therapy of fractional laser with bit application of Ethocyn. It was an 8-week trial. His patients were 28 to 62. Again, we'll see these. This is just an example of a patient. He did the study on half-face, and it wasn't a complete half-face because women weren't going to undergo a laser procedure on half their face. What he did was on half their face, they had laser on the entire face, and half the face they were allowed to use Ethocyn. And just in the frontal area, it counted the ear growth. And those that had used Ethocyn were markedly better looking, showed improvements, had smoother, tighter skin. These are biopsies from Dr. Hogan's trial. This is his publication of that study with the laser. It was last September in Colorado. This is Dr. Hogan's, Dr. Echevarria. He's an eminent dermatologist. This is in Peru. This was August of last year. And this is the ageLOC formulation. Okay, I need to take a break.